Hello, we would like to welcome you to the webcast entitled Biosimilars, a new era of gastroenterology management considerations. This is part 2, Biosimilars and Patients with Inflammatory Bowel Disease. This educational program is provided through an educational grant from Pfizer Incorporated, awarded to the American Society of Gastrointestinal Endoscopy, the Society of Gastroenterology Nurses and Associates, and the France Foundation. My name is Mike. I will be your operator for our presentation today. Now, before we get started, I would like to take a moment to acquaint you with a few features of this web event technology. At any time, you may adjust your audio using any computer volume settings that you may have. On the right-hand side of your screen, you see the Q&A window. There's a large window which holds all of your sent messages and a smaller text box at the bottom where you can type in your questions. To send a question, click in the text box and type your text. When you're finished, click the Send button. Your questions will be answered as time allows and will be addressed during the Q&A portion of today's event. Please note your attendance will be marked and provided to the France Foundation. Should you experience any technical difficulties with the web portion of today's program and need any assistance, please call our technical support line from Europe, dial +49 699 675 8428. All other countries dial 1-858-345-5913 or contact us at livewebcast.net/support and an operator will dial out to you. We are pleased to present our 3 faculty members for our program this evening. Kenneth McQuaid, MD, is a practicing gastroenterologist in San Francisco, California. Edward Lee, PharmD, is a professor in the Department of Pharmacy Practice at the University of New England College of Pharmacy. Catherine Bauer, MSN, RN, is the Director of Endoscopy at the University of Virginia Medical Center. In today's presentation, you will hear their expert perspectives on the use of biosimilars in the GI clinic. This live webcast is available for CME credit from ASGE and for CNE credit from SGNA. You'll receive instructions on how to claim credit at the end of the program. This activity is being recorded and will later be available online for those who missed today's broadcast. Please note that all financial relationships disclosed by the faculty have been resolved in accordance with the ACCME standards for commercial support. And at this time, I'd like to share with you our activity learning objectives. The event today will last approximately 1 hour, and at this time, I'd like to turn the microphone over to Dr. Edward Lee to begin the presentation. So, Dr. Lee, welcome.
Thank you for that introduction, and thank you, everyone, for joining us tonight. And it certainly is an exciting time to be talking about biosimilars. I've been talking about biosimilars since 2011, when it was just an abstract concept that was just being formulated at the FDA level. And it's exciting today in 2017, or at the end of 2017, that we have a number of biosimilars approved in the United States, starting with filgrastim biosimilar in 2015, but you can see that we've recently had 3 new biosimilar approvals in 2017. So today we have 7 products that are FDA approved, 7 biosimilar products that are FDA approved in the United States. And when we're specifically talking about the gastroenterology space, you can see that we have 1, 2, 3, and 4 products specifically approved in the gastroenterology space. So these are Infliximab biosimilars and adalimumab biosimilars, which we know to be extremely expensive products and ones that show up at the top of the list of overall and total expenditures in the United States. So our healthcare system is spending an enormous amount of money on these products, and so it's great now to have some competition in the space, which will hopefully moderate some of the spending on these products, and also hope to increase access to these products because of the increased competition. And so the reason why we're here today is to talk about the differences between biosimilars and generics and the differences between biosimilars and reference products, because the regulatory approval of biosimilars is different than a reference product or a generic. And recall that a biosimilar from a regulatory standpoint has to demonstrate that it's comparable or that it's highly similar, so that's standard number 1, with no clinically meaningful differences to the reference product. So that's standard number 2. So the reference product already demonstrated that these molecules are safe and effective, and so the exercise is one of comparability. And the highly similar standard is based on robust analytics, so looking at the structure and function of the molecule. This is analogous to being identical to so a generic product, a generic small molecule is identical to its reference, and for a biologic, being highly similar is essentially analogous to being like another batch or another lot of the reference product. So it has to meet some very, very strict standards to be deemed as being highly similar. And so this analytical exercise forms the foundation of the biosimilar exercise. At the same time, the biosimilar company needs to demonstrate that their product is not clinically meaningfully different based on targeted clinical trials in a sensitive population. And what that means is that if there was truly a clinical difference between the biosimilar and the reference product, then that would be picked up in this particular study using this specific sensitive population. So with that, I'd like to turn it over to Kathy, who's going to walk us through a few cases. So Kathy, take it away.
Sure. Our case number 1 is 26-year-old female with ileal Crohn's disease. She was diagnosed 6 months ago and initially treated with budesonide with good symptom relief. Symptoms reoccurrence after steroid tapering, though. Subsequent treatment with azathioprine, no symptoms improvement despite adequate metabolite levels. So current presentation, she's got poor energy, cramping, abdominal discomfort, 3 to 4 loose stools per day, and perianal discomfort. On physical exam, she's got mild right lower quadrant tenderness, no mass, large perianal skin tags with anal fissure. Her labs are right there, hemoglobin 11.2, white count of 12,000, albumin 3.1. She has normal liver chemistries, and CRP is 15. Her colonoscopy a week ago, multiple erosions and superficial ulcers in the distal 15 centimeters of the ileum without stricturing. Her colon was normal. The following discussion, a decision is made to initiate treatment with Remicade, 5 milligrams per kilogram at weeks 0, 2, and 6. We followed by maintenance therapy, if effective. We have a question. Is therapy with anti-TNF a reasonable approach at this stage? And you can click in and vote your response to that.
Just a reminder for all the attendees, you simply click on the radio button to the left-hand side of the response you feel best represents this response to this interactive polling question. Kathy, I'll turn it back to you.
Are you going to broadcast that?
I will, in fact.
We're getting a lot of yeses so far. How does that look, Dr. Lee?
That looks great.
We've closed the voting booth, and majority of you said yes.
Yeah, so this is Dr. McQuaid, and I think the way people answered is consistent with what most gastroenterologists would do at this time, although this remains a little bit of a controversial area. Actually, the AGA, American Gastroenterological Association, back in 2014 published its guidelines on the use of biologics, and in that guideline, they asked that you initially look at patients and try to identify whether they appear to be a patient at low risk for sort of progressive disease or at moderate to high risk. This patient actually had a few risk factors for disease. Namely, she was less than 30 at the time of her diagnosis. and she has now a perianal disease, which is a more worrisome feature for progressive disease. Now, on the favorable side, she had somewhat limited disease. It was ileal disease only, and so far has not had stricturing on her colonoscopy or a penetrating disease, that is a fistula. But nonetheless, I think the fact that she has recurred after a course of budesonide and an immunomodulator, azathioprine, that her symptoms have come back, and that she has perianal disease and is young, she would qualify as a moderate to high-risk patient, and we would therefore favor, at this time, putting her on an anti-TNF drug. And infliximab or adalimumab would both be reasonable. And so this, again, she, according to the guideline published in 2014, they would favor for the moderate to high-risk patient. And let me just kind of point the arrow here. For that moderate to high-risk patient, that you would use anti-TNF therapy with or without an immunomodulator at this time. And we'll talk about that in just a second. I would favor not using an immunomodulator, but we'll talk about that coming forward. And so, Kathy, I'll turn it back over to you for some further questions.
Sure. So continue with the same patient. We're going to, we heard that the patient is notified that her insurance company will cover the biosimilar infliximab rather than the reference product Remicade. So prior to her scheduled infusion, she called the practice nurse as she is concerned that she may be receiving a less effective therapy. What would you say to this patient? So our question is, which infliximab would you recommend for this patient? Remicade, Inflectra, Renflexis, all of the above are reasonable options or unsure?
Again, just that reminder, the radio button next to the response that best represents how you feel about this interactive polling question.
It looks like we've got all of them in. And the majority of you say all of the above are reasonable options. So Dr. McQuaid, how would you answer that?
Yeah, so this is the real question that Ed has laid out for us initially here. We have these biosimilars that are on the market. Here we have a patient that's not been on a biosimilar or been on a biologic agent before. And the question is, do we insist upon the Remicade, the Innovator product, or are we comfortable with going with the biosimilar options? My own feeling is that in this particular patient, that answer D, all the above are reasonable options, is a reasonable thing. That for patients who have not previously been on a biologic agent, and your choice is between the Innovator agent or the biologic agent, the biosimilar agent, that the biosimilar is a perfectly reasonable option. And I'll turn it over to Ed to discuss here how we feel, how it is that we feel comfortable with using biosimilars here, and he'll talk about the data that the FDA requires before allowing these agents to be used clinically.
Right. Thanks a lot. And the big question is always revolves around the data, and what data really demonstrates that we could use all of these. And again, I need to add the caveat that with a biosimilar, we're not assessing safety and efficacy of the molecule of infliximab. That's already been demonstrated by the reference manufacturer, by the Innovator manufacturer. So again, the exercise here is comparability. Is manufacturer B and C, is their product of infliximab equivalent to manufacturer A, which is that Innovator product? Okay, so when you look at the data here, you see that in this trial in rheumatoid arthritis patients, that both of the infliximab biosimilar products, so that's CTP13 on the left and SB2 on the right, When you look at their ACR20s at week 30, the response rate and the difference in response rates, you can see that those were very, very similar to each other. Actually, when you look at the statistics with that, the confidence interval needs to fall between a predefined margin, equivalence margin, and for both of these studies, they comfortably fit within their predefined equivalence margins for a statistical similarity to each other or comparability to each other. So in these two large-scale studies, what we would consider to be kind of similar to phase 3 randomized controlled studies, we don't call them that for biosimilars. We call them clinical comparative studies. But in these large-scale studies, it showed this equivalence in rheumatoid arthritis patients. And I know what you're thinking, which is, well, why, that's rheumatoid arthritis and that's nice, but how do we get to the gastroenterology indication? So remember that the goal of the regulatory paradigm, approval paradigm, is comparability. So what we do is we utilize a concept called extrapolation that's based on all of the data that we know about the reference product, all of the data that we know about the biosimilar product, and look at and extrapolate as to whether or not these other indications that the reference product has can be given to the biosimilar product as well. So what does this look like? So first, we take a look at the Biosimilar Infliximab, and we look at the analytical studies, the non-clinical studies, and the PK studies as well. And remember, the analytical studies that show that the biosimilar is highly similar to the reference, that's the foundation and incredibly important. So if, based off of these analytical studies, the biosimilar can be essentially considered to be another batch of the reference product, in terms of these analytical features, that's a really, really good thing. And so our concern about whether or not we would be able to use the biosimilar in all of the different indications is really, really, we don't really have many concerns about that. Also, if we look at the PK studies and we see that the biodistribution is the same across different models, then we don't have many concerns as well. So then, if we look at these confirmatory clinical studies in a sensitive population like rheumatoid arthritis, where if a difference existed, we'd pick it up in this patient population, and we see that they're equivalent, then we can look at the totality of the evidence, what we know about the biosimilar, what we know about the reference, look at how the structure and function of the biosimilar molecules compare to the reference, look at things like biodistribution, and feel confident that we can extrapolate that to all of these other indications that are listed here as well. So this is actually a regulatory concept that the FDA talks about. It is part of the FDA process. It isn't just made up out of thin air. And it is what they call scientific justification. So this needs to be a pretty rigorous exercise to justify this extrapolation. So I think at this point, I'll kick it back to you, Kathy, to ask the audience what they think.
Yeah, so I had a question for you first. So it sounds like it would be pretty easy to explain to the patient that there is similarities and the effect of both of those would be the same, whether it be Remicade or the biosimilar.
Yeah, absolutely. So both of them have, both of the biosimilar products have similar types of data demonstrating that they're comparable to the reference product. So either one of those, you could, tell the patient either one of them would be okay. Okay.
So our question: can you have confidence in the safety and efficacy of Biosimilars Infliximab for a patient with Crohn's disease, which was not one of the indications studied to support the biosimilar approval? And it looks like we've got voting already. So it looks like the majority of you say yes, totality of the evidence supports efficacy and safety and conditions of use for which the reference product is licensed, which is good. We'll take it to the next slide.
So, so this is Ken McQuaid again. And I actually understand why there's roughly 20% of people that are unsure, because as Ed said, rheumatoid arthritis is not necessarily the same as Crohn's disease. And while the people at the FDA are very smart people, still we'd like to see some data done in IBD. What data do we have? Well, we do have data. This was actually the Norwegian IBD study, in which 78 patients made up of ulcerative colitis and Crohn's disease patients, 32 and 46, with moderate to severe disease despite steroids, so similar to our patient, were either given we're given the biosimilar product and looked at after 6 weeks of treatment, and they looked at endpoints for remission using for Crohn's disease the Harvey-Bradshaw score, or for ulcerative colitis the Mayo score, and they also measured CRP levels, leukocal-protectin levels, and the trough levels of the infliximab, and as well as anti-drug antibodies. So, a study that reflected a real-life clinical practice, people with moderate severe disease, not getting better on steroids, put on a biosimilar product. And when we look at the upper left corner, the Harvey-Bradshaw scores in patients with Crohn's disease, or on the upper right-hand corner, the Mayo scores in patients with ulcerative colitis at week 14, which is where we often will assess disease remission. You can see that significant proportion of the patients dropped their Harvey-Bradshaw scores under 4, and their Mayo scores went down as well. And overall, the number of patients in clinical remission was 56% in Crohn's disease, 79% in ulcerative colitis, which is pretty consistent with what we've seen with anti-TNF agents in the previous trials using the Innovator product, Remicade. When we look at C-reactive protein in both ulcerative colitis and Crohn's disease, again, these fell, showing that going along with the clinical Harvey-Bradshaw and Mayo scores, the inflammatory markers went down as well. So we do have data done in IBD patients that show similar rates of remission, similar responses of inflammatory markers, as has been seen with the Innovator product. And I think that in a patient, as we're discussing here, who's going on a biologic for the first time, that we can feel comfortable that the biologic biosimilar is every bit as effective as the Innovator product, in this case, Remicade. And overall, now there's over 20 different publications and abstracts looking at biosimilars, infliximab in IBD, done throughout Europe and Asia, with more than 1,700 patients treated with one of them, the CTP13. And there have been both induction and maintenance studies with follow-up now up to 54 weeks, showing that overall, when you look across the publications, there's consistent responses at remission rates of 50 to 87% for Crohn's and 37 to 100% with ulcerative colitis. I think that overall, the data are strongly supportive that, just as we saw in the randomized control trials done with in patients with rheumatoid arthritis, that we're seeing a large number of induction and maintenance studies done in patients with IBD that show excellent remission rates. I feel comfortable initiating a biosimilar in patients who haven't been on a biologic before, as was done in our patients. So, Kathy, I'm going to turn it back to you.
All right, so we're going to start our treatment with Biosimilar Infliximab at 5 milligrams per kilogram. Some additional questions that we have: should infliximab trough levels be obtained? And if so, when? And should the biosimilar be given as a monotherapy or in combination with azathioprine?
So this is an article that just came out, again, from the American Gastroenterological Association, published in Gastroenterology just in August, which actually addresses this very question, which is how and when we should monitor drug levels of infliximab or adalimumab, and whether we should be using immunomodulators with the infliximab or adalimumab as well. And in the guideline, what it came away with was that in adults with active IBD treated with anti-TNF agents, they suggested that drug monitoring being done in order to guide treatment changes. That is, in a patient that's on these drugs and either is not responding or has lost response, that it was reasonable, with a conditional recommendation, to check the trough levels of the infliximab or the adalimumab and to assess for the presence of anti-drug antibodies. They suggested that reasonable trough levels for infliximab were greater than or equal to 5 micrograms per ml, and for adalimumab greater than or equal to 7.5 micrograms per ml, based on studies that were out there. And so what they really were endorsing was they felt there was enough data to support what they're calling a sort of a therapeutic drug monitoring, that is, using it to guide treatment changes. But in our patient, who is just initiating treatment for the first time, they wouldn't necessarily recommend that drug levels be done if she was responding well to the biosimilar product. And that is an area, I think, of ongoing controversy. The AGA, through its guideline, would recommend that if she wasn't doing well by week 14, that drug levels be done to see if she had adequate levels, and we'll go over that in just a second. But left open is whether we should be checking drug levels if she is doing well. Many people, including myself, would check drug levels because there is a belief that maintaining a trough level above a certain level may reduce the risk that she will develop anti-drug antibodies, which we would prefer to avoid. And we perhaps would prefer to maintain trough levels that are sufficiently high in hopes that we don't wait for her to develop sort of clinical manifestations of active disease with potential stricturing or fistulization. That is, we'd like to sort of preempt problems down the line by maintaining adequate trough levels. However, as I say, the AGA guideline didn't make recommendations on whether trough levels should be obtained in the patient that's doing well. So in this particular patient, she's been initiated on the biosimilar product. As I say, many of us would check trough levels at 14 weeks, even if she wasn't doing well, but the guideline would favor that you do trough levels at 14 weeks or potentially even earlier if she is not doing well. Now, how do these help you is shown in this figure here. And that is that if you measure the anti-TNF levels and the anti-drug antibodies, and I will bring the arrow down here, and she has anti-TNF, she has undetectable or low TNF levels. So in other words, we check it and she has low infliximab levels, less than 5. And if she does not have anti-drug antibodies, she just doesn't have enough drug on board. And we would recommend that her dose of infliximab be increased to either by shortening the interval between the infliximab doses to say every 6 weeks or every 4 weeks, or by increasing the dose to say 10 milligrams per kilogram. So if she's not doing well and has low anti-drug levels and no anti-drug antibodies, everybody would agree that we should increase the dose of her infliximab. If she's not doing well and she has high infliximab levels, or high anti-drug antibodies, then if she's got high infliximab levels and is not doing well, then she's not responding to the drug. And so there again, if she's got a level that's more than 5 or certainly more than 10, and she is not responding to the drug, she's not going to respond to an anti-TNF agent. And the recommendation in that case is, rather than switching to another anti-TNF drug, to switch to a different class of drugs, say something like ustekinumab or vedolizumab, because in this case, if the patient hasn't responded and has adequate drug levels, then she's not going to respond to these anti-TNF agents. Now, if she has undetectable or low infliximab levels and has high anti-drug antibodies, in that case, she has now antibodies to this infliximab, and she's not going to respond to this infliximab or any other infliximab product. In that case, it doesn't necessarily mean she's not going to respond to a different anti-TNF drug like adalimumab, and you could consider then, if she has high anti-drug antibodies and undetectable infliximab levels, that you would switch to a different anti-TNF drug, say something like adalimumab. On the other hand, if she has undetectable levels of the infliximab and low anti-drug antibodies, well, then they're suggesting many of these low anti-drug antibodies will go away or are transient and fluctuate back and forth and are not necessarily there in sufficient levels to be neutralizing the drug. In that case, you could actually consider going up on the infliximab dose or adding an immunomodulator to see if she would then respond with more adequate levels of infliximab. So long and short of it, in patients not doing well on the infliximab drug, we would typically get trough levels of the infliximab and measure the anti-drug antibodies. If the trough level is low and there are no anti-drug antibodies or they're low, we go up on the dose of the drug. If the trough levels are adequate or high, it means she's not going to respond to the infliximab, and we should go to a different class of drugs. If the trough levels are low and she has high anti-drug antibodies, you could consider going to a different anti-TNF drug like adalimumab. If the trough levels are low and she has low anti-drug antibodies, they may be transient, they may go away if you increase the dose of the infliximab, and you could go up in infliximab and see how she does, or you could add an immunomodulator, which may also affect the presence of these anti-drug antibodies. So I hope that's clear. It's an excellent review on this subject. Was published in Inflammatory Bowel Disease in 2015, as cited in the figure here. So with that, I will turn it back for just a discussion here for how people feel about this. My own perspective is that, I would start her on the infliximab at the doses that we listed. I generally would, even if she was doing well, check her infliximab trough levels at 14 weeks, but that's not uniformly recommended. I certainly would check her infliximab levels and her anti-drug antibody levels at 14 weeks if she was not doing well. And I'd be curious to see Kathy and Ed how you feel about that.
Yeah, I would be concerned with her confidence level, but with all of the data and information in front of her, she should be fine with the biosimilars.
Yeah, exactly. And kind of my perspective on this is that it's all the same depending, regardless of if they're getting the biosimilar versus the reference product, and you would treat each of those products, Remicade, Inflectra, and Renflexis, as infliximab, and the algorithm would be the same for any of them regardless.
So we have a case.
Okay, I think that brings up another case.
Yeah, case number 2. We have a 39-year-old man with a history of severe ulcerative colitis. He was diagnosed 3 years ago. his entire colon involved. He had a short hospitalization with IV corticosteroids. As an outpatient, he had colitis flared with prednisone and was tapered below 20 milligrams per day, but unable to tolerate mercaptopurine, but hypersensitivity. 2 years ago, initiated Remicade, complete clinical remission, and sigmoidoscopy 1 year ago confirmed mucosal healing with a Mayo score of 0. His infliximab maintenance, 5 milligrams per kilogram every 8 weeks. Trough level 6 months ago was 8. No anti-drug antibodies. At his most recent infusion, the patient was informed that his health plan wishes to switch him to a biosimilar infliximab going forward. The patient calls a GI nurse specialist with concerns about this proposed change in treatment. What should the nurse tell him? Question: is a switch to a biosimilar reasonable for this patient with a history of UC who has been in stable, prolonged remission with Remicade? Vote. Sounds like everybody's in favor. Switching. Unsure. So, Dr. McQuaid, we have a couple that say no, and the majority yes.
Yeah, so I think this is a real conundrum, frankly, and I suspect perhaps many of us would agree to disagree. Here you have a patient that had pretty severe ulcerative colitis that didn't respond to corticosteroids and mercaptopurine, but now has done beautifully on Remicade, and somebody wants to switch, at this stage, that switching does require notification of the physician and the patient, as was done here. There would need to be an informed discussion with the patient because, it's going to be very hard for patients who are, doing well on this lifeline of a drug to perhaps reassure them that switching them does not pose any risk to them of flare or otherwise. And so what data do we have to reassure them? Well, there was this large study done in Norway, a phase 4 study, in which over 400 patients with different conditions, including Crohn's disease and ulcerative colitis, 155 with Crohn's and 93 with ulcerative colitis, were randomized, and they were doing well. They'd been stable on their Remicade for at least 6 months. So they were either in, a clinical remission, stable patients, randomized to either continue the Remicade or to switch to the biosimilar Infliximab, the CTP13, and followed for 52 weeks. And overall, when you looked at the 52-week follow-up, the number of patients, and let me get my arrow again here, that were it showed disease worsening during the 52-week follow-up was 26% with the Remicade and 29.6% in patients switched to the CTP13 product. And that difference of 4.4% had a confidence interval of anywhere from minus 12% to plus 13%, so it was not clinically or not statistically significant. And overall, the number of people who remained in remission were 61% with the Remicade and 61% with the CTP13. So in this large study, over 400 patients, half randomized to the biosimilar product, half remained on Remicade. No difference in the remission rates, no difference in number of patients losing response when either on the original product or the switch to the biosimilar. There was also no difference in the number of patients developing antibodies. This has been a concern. If you switch the patient from one product to the other, Ed went over the fact that these could be manufactured slightly differently. There's always been a concern. Could it in some way have a difference in terms of its likelihood of developing antibodies? But at least in the number of patients that developed anti-drug antibodies over these 52 weeks, it was no different in those patients who continued on the Remicade versus those patients who switched to the biosimilar product. And so my own feeling is that I understand why patients would have concerns about switching when they've been doing very well on one product. But the data that we have out there, the published data, suggests that the likelihood they will remain in remission is the same, whether they remain on the originator product or switch to the biosimilar product. This has been corroborated in another study that was published here in June in the Disease and Sciences, which was a single-center study of patients with IBD who were switched from the Remicade to the biosimilar product and followed over the year. And there was no mean change in the patients with Crohn's disease in their Harvey-Bradshaw scores. There was no change in their ulcerative colitis scores, no change in their fecal calprotectin levels or CRP levels, and no significant increase in the number of patients developing anti-drug antibodies. So we have this, a growing body of data, both randomized control data and cohort data, suggesting that patients who are switched from the originator products to the biosimilar products do equally well after switching. Nonetheless, it's an issue that needs to be discussed carefully with patients because they're the ones that have the disease, and when they're doing well, are going to have a lot of concerns about this switch. We need to provide them the data that we have available, and help and participate with them in the decision-making process. So with that, I'll turn it back to you, Kathy, to continue with this case.
All right. So we're switching the patient to biosimilar infliximab maintenance treatment. Additional questions that might be asked: should dosing and frequency be the same as with the reference product? And should trough levels be obtained to confirm adequate drug levels? If so, when? Should co-therapy and methotrexate be considered to reduce the risk of immunogenicity?
Right. So this is come back to the questions here. The dosing is identical to the reference product. It's not any different. And whether trough levels should be attained is what was discussed in the previous case. This person was doing well on his Remicade before. We've switched into the biosimilar product, and we would expect, therefore, him to have similar, response rates. So the question of monitoring for trough levels and anti-drug antibodies comes back to that guideline I mentioned from the AGA that was just published. And they would say that if he's doing well and remains in clinical remission, that they would not necessarily recommend that he have trough levels done or anti-drug antibody levels done. Again, I think for many of us out there, depending on the patient's insurance status and whether that insurance company will pay for trough levels, we do favor periodically measuring trough levels in patients, even if they are doing clinically well. And I think that in a patient that's been switched from the Innovator product to the biosimilar product, I personally would prefer to check a trough level at some point after switching, partly to reassure the patient as well as myself that their trough levels remain adequate. But again, the guideline would only say that there's conditional recommendation that trough levels be done in patients that are not responding or have lost response. As far as adding an immunomodulator with the drugs, the guideline from 2014 says that using an immunomodulator with a biologic agent is recommended in people that are on their second or third biologic, but not necessarily on their first biologic. So since this was the first biologic he was on, provided we maintain good trough levels, the likelihood he will develop anti-drug antibodies is low. And that's the main reason that we start the immunomodulators, especially methotrexate, was to reduce the risk of developing an anti-drug antibody. So personally, on this 39-year-old man, if he was stable and having a good continued maintenance of remission on his biosimilar product, I would check a trough level at some point and verify that it's still adequate. I would not be initiating a co-therapy with an immunomodulator agent like methotrexate. I would only consider that if he lost response and needed to go on to a different anti-biologic agent. And I can turn that back over to Ed to see if he disagrees with that from a pharmacy standpoint.
No, I think those are spot on, and I definitely agree with those recommendations. And I want to pivot a little bit because we have this question about cost from the audience, which asks about the fact that development costs are lower with biosimilars, and presumably, we think that that's going to translate to kind of reduce costs to patients, and the audience member is kind of asking if that's the case. And I think when we're considering the switch from the reference product to the biosimilar, the biosimilars on the market today, their list price is lower than the reference product. So something called AWP, average wholesale price, or average sales price. That list price is lower than the reference product. Now, does that mean that each individual practice is going to pay that? No, that's kind of like the MSRP of the drug world, where it really depends on your specific contract, at what price you're going to get things at. But, overall, they're lower. And what that translates into, kind of when a patient switches over from the reference to the biosimilar, what that could translate into is a lower copayment amount. So if a patient is paying a percentage of coinsurance, then a percentage coinsurance off of the cheaper product is going to be a lower copay. The other thing to think about too is that I think we have a lot of people on patient assistance programs from the reference product as well, and so these help with the copays. And when we're considering switching over to a biosimilar, it may be worthwhile to look at the different copay assistance programs that the biosimilar companies have to offer, and see which ones the patients would qualify for.
That's a great point, Ed. So if we go to the next case, we have a 35-year-old man with Crohn's disease, diagnosed 8 years ago with CD involving distal ileum and right colon. We tried various diets and alternative therapies. Symptoms progressed, and he was placed on Humira for 6 months. 1 year ago, small bowel obstruction that required resection of the cecum and 35 centimeters of the ileum. Postoperatively, he was placed on Infliximab 5 milligrams per kilogram every 8 weeks. Currently feeling well, routine colonoscopy performed, and extensive ulceration in distal 15 centimeters of neoterminal ileum without a stricturing. Infliximab trough levels unmeasurable, and high anti-drug antibodies. Should this patient be considered a failure to anti-TNF therapy? Right, it looks like we've getting close to the end there. Majority say yes. And the next question, as the patient has developed antibodies to Remicade, should a biosimilar Infliximab product be considered? Going all over the board, huh? Feels like election night. So it looks like majority are saying no because the anti-infliximab antibodies will cross-react with biosimilars. Is that the case, Dr. McQuaid?
Yeah, so this is a good question, and I think the audience is mostly going to question. So first of all, is he a failure to anti-TNF? Not technically. He's developed antibodies to the adalimumab, but that doesn't mean that he wouldn't respond to a different anti-TNF drug. So, but we wouldn't give him a biosimilar adalimumab because if he's already got antibodies, and we'll go over this, to one adalimumab, they will cross-react with whatever biosimilar you give. So is he a failure to anti-TNF? No, you might switch him to Infliximab, and he might do okay with that. If I did switch him to Infliximab, I would certainly put on that with an immunomodulator because I would want to really reduce the risk of him developing antibodies. But he isn't technically a failure to anti-TNF. He is a failure to adalimumab because he's developed antibodies and should not be put on a biosimilar product. And in this study done in gut in 2016, this is just to show that among patients that had, and again, I'll bring this down here, the pointer, there we go, that among patients who had anti-Remicade antibodies in this case, that there was a similar level of inhibition with of the antibodies to both Remicade and the biosimilar product. And so we have no reason, once you develop antibodies to one, the more than likelihood they're going to be cross-reacting with another drug from that same group of drugs. So if you've got antibodies to Remicade, they're going to respond to the biosimilar Infliximab. If you've got antibodies to Humira, they're going to react as well against the biosimilar adalimumab. So in this gentleman, should a repeat trial of adalimumab be considered? That's the question. And I don't think we have a polling question on this. Yes, no, yeah. So the same question really as with the Infliximab. He had antibodies to this adalimumab. We would say a repeat trial of adalimumab should not be given. If you have because I think that was the question of this gentleman. He was on adalimumab to begin with, if I have this correct. Is that correct? I take it back. I'm sorry. He was on the Remicade. So could he go on to the adalimumab? Yes, he could go on adalimumab, but he should not go back on a biosimilar product for a Remicade. But could he have a repeat trial of adalimumab? Absolutely. Just because he had antibodies to Infliximab does not mean he'd have antibodies to adalimumab. And so going on to adalimumab would be a reasonable option for him. And this is just a study done for the Annals Rheumatology, looking at biosimilar adalimumab versus the Humira, showing that in patients, again, with rheumatoid arthritis, that they had similar responses using the ACR20 measurement tool with both the biosimilar product and the Humira, the Innovator product. So these drugs, again, seem to work equivalently, both the Humira and the biosimilar product. And it would be a reasonable thing in this gentleman who developed antibodies to Remicade to switch to a different anti-TNF group, in this case, adalimumab, and expect that he would respond. And I personally, if I did that on him, since he already developed antibodies to one anti-TNF drug, I would do this in co-therapy with an immunomodulator like methotrexate or like mercaptopurine to further reduce the chance that he would develop antibodies to the adalimumab product. And I think we're getting near the end of the time here, but just as we had the studies done in rheumatoid arthritis, we have studies done in plaque psoriasis showing equivalent response rates of adalimumab with the Innovator product, Humira, as well as the biosimilar products. So let's go back to this case here, Kathy.
Sure. So additional considerations if anti-TNF therapy is continued, should co-therapy with immunomodulatory agents be given to reduce the risk of antibiotic formations? Or should a trough level be obtained, and how is this patient's disease actively best monitored going forward? Right.
So again, because he already developed, he's already had surgery. He's had, so we'd like to really, have this man avoid another surgery in his future if possible. He's now developed antibodies to the infliximab, so we know we can't give him that group of drugs. He's now switching to adalimumab. I would feel fairly strongly in him that we would like to give him a co-therapy with either methotrexate or another immunomodulator like azathioprine or mercaptopurine to reduce the risk of him developing antibodies. I would also favor checking drug levels on him, both because I would like to have adequate drug levels in the hope of preventing disease recurrence, but also because maintaining adequate drug levels in the case of adalimumab above 7.5 reduces the chance that he will develop antibodies to the adalimumab. And so I would favor in him that once I had him on the adalimumab, that I would check trough levels of the adalimumab, anti-drug antibodies, and verify that I have adequate trough levels. And I would probably monitor his drug levels approximately once per year. And turn it over to you, Kathy, from the perspective of a GI nurse, and Ed, to see whether, how you feel about that in this gentleman.
It sounds doable for us to explain to the patient the needs and all of the continuing monitoring. I think we can justify that with the patient.
Right, absolutely. And, the therapeutic drug monitoring, I think, is been a really great thing, especially in this person with a personal history of that, with issues related to antidrug antibodies. I think that is all the more case to do more intensive monitoring.
So we'll go to our last slide.
Yeah, I was going to say, this remains an area of controversy, certainly, but I think most people are using the anti-drug antibodies and trough levels more and more aggressively now to monitor patients. We know that if we maintain levels above a certain trough, and those levels given earlier were 5 for infliximab and 7.5 for adalimumab, that we appear to reduce the chance of developing anti-drug antibodies and appear to increase the likelihood of maintaining remissions in patients. That's clearest that these measuring both is of value in patients that have failed to respond or have lost response, but many of us are also measuring them in people that appear to be in clinical remission and following them long term. We'd like to maintain adequate levels at trough levels to reduce the chance of developing anti-drug antibodies and try and maintain them in remission.
So the key message is the increasing availability of biosimilars has the potential to broaden patient access to biologic therapies and provide cost savings for patients with IBD. An approved biosimilar can be expected to have the same efficacy and safety profile as the reference originator product in the approved indications. If a patient is doing well on a reference product, similar efficacy and safety can be expected with a switch to a biosimilar. Patients who have developed drug antibodies to a given biologic should not be switched to another version of the same biologic. Okay. And members of the healthcare team need to be prepared to answer patient questions about biosimilars, as patient education will be important for optimal incorporation of biosimilars into clinical practice, and that's really key to nurses to know the reactions in the patients and the questions that they'll have. We need to be dedicated to them.
And I think in this scenario, we had the 3 different cases to amplify those points. The first being a patient that was starting a biologic, and we agreed that starting a biosimilar was a perfectly reasonable choice. The second was a person who was stable and now is being notified that the company would like to switch him to a biosimilar, and we presented the data that suggested that that's a reasonable option, but we needed to have a discussion with the patient to make sure they were comfortable with it. And the third being a patient who had had surgery, now has a recurrence of their disease, and on drug monitoring is found to have high anti-drug antibodies, and we agreed that we would not keep that person on that biologic, in that case infliximab, but that it was reasonable to switch to a different anti-TNF drug like adalimumab, be it the Innovator drug or the biosimilar, and follow them more closely for trough levels and anti-drug antibodies.
Very well. Just a quick reminder to the attendees, we have just a moment or two for your questions. If you want to participate in the Q&A portion of the event, we encourage you to go ahead and ask your questions. You can write those in the Q&A window to the right-hand side of your screen. Send a question, click in the text box and type your text, and when you're finished, just click the send button. If you are connected via the telephone and you'd like to call in for your question, you can just simply press *# to queue up with a question. And with that, Ms. Bauer, I'll turn things over to you. It looks like we may have at least one additional question, and we'll see what else might transpire.
Yes. So a question, in the third patient, he appeared to have a reoccurrence and needs surgery on Humira. Would it be appropriate to switch classes entirely, such as Stelara or Entyvio?
Right. So I think I stumbled on that one. His initial drug was Remicade, but I think that so it is, and he had high anti-drug antibodies. I don't think that he necessarily had failed anti-TNF, so the options are really to switch him to a different anti-TNF drug like Humira, or to switch drug classes. In this case, going to ustekinumab or vedolizumab, and I think either is a reasonable option. I think that if, as I say, if you switched his anti-TNF drug, I would follow his drug levels carefully, his trough levels, and I would follow him for anti-drug antibodies, and I would want to look at his response rate with another colonoscopy and or some sort of a MR enterography study to verify that he had shown a clinical response to the switch in anti-TNF drugs, and to verify that really his ulcerations were healing. Because what you don't want this man to do, who's already had one surgery, is to go on to stricturing again or fistulization and require another bowel surgery. And so I would want to look at, if I kept him on anti-TNF agents, I would want to look at a reasonable time frame, say 10 to 14 weeks, to see whether healing had occurred. It would also be reasonable, however, to switch him to ustekinumab or vedolizumab. My own experience is that in dealing with the drug companies, that many of them will insist in a patient like this that we try a second anti-TNF drug before we switch drug classes. And I believe that's largely driven by cost considerations. So a lot of this will be determined by what drug companies will allow, what your pharmacy plan will allow. But I think either way, it would be a reasonable thing to do, switching the anti-TNF drug or switching drug classes entirely and going to ustekinumab or vedolizumab.
All right, everyone. And with that, seeing no additional questions and no phone questions at this time, I don't know if any of our presenters have any concluding thoughts you'd like to share with the audience before we wrap things up today. And hearing none, with that, we'll go ahead and reach the end of our program. On behalf of ASGE, SGNA, and the France Foundation, we would like to thank you for participating in today's event. Once the event has ended, you will be redirected to claim your CME or CNE certificate. If a window does not open automatically, please click on the link that says click here to take survey in order to proceed. This will conclude our webcast for today. Thank you, and everyone have a great rest of your day.